So sorry.
La caméra, tu veux la tourner?
Ouais.
Okay, thank you for being there for the second session of our Brazilian French seminar on the history and philosophy of science. This afternoon, the first speaker is Jean-Baptiste Grodwohl from the University of Paris-Cité in Paris, of course, and he is one of the better specialists of the history of population genetics. He's working on those topics for more than 15 years, I guess, now, maybe a little less. Okay, but...
I'm younger than you.
Well, yeah, but not that much. The title of this presentation is on the history of population biology, from ecology to population genetics.
Obrigado. I had a very productive talk with João yesterday, and I decided to give a different lecture than the one I planned. I will try to sell him history of science rather than philosophy of science. So everything I will say will target it at him. So I hope you accept to be part of the experiment. I have worked on the history of science in the 20th century, and many of the sciences we call part of population biology, that is animal behavior, population ecology, and population genetics. And my main focus is on the history of these sciences in the English-speaking world, and mostly on the United States. For the topics that were of interest to me, I felt that this was where the most informative developments were. This being said, I don't think that the history of science, especially contemporary sciences, should be limited or restricted to the English-speaking world. There are extremely strong incentives that can lead us to believe that everything important is being done or has been done by American scholars, and that the rest is less important. I think we need to react to this. Our responsibility as historians is to pay attention to the complexity of geographies and histories. And in other words, through these exchanges, I hope to learn more about the history of science in Brazil. And if we can talk, I hope that can foster research and our interest in the history of science in Brazil. There is so much that we in Europe, we don't know about the history of sciences in Brazil. And there is so much that you can offer. So I'm sure that Darwin is a fascinating subject, and many studies on Darwin for centuries, but do not forget the unique things you can bring to the world. I will say a few words about, in this presentation, I will say a few words about my own work as a historian of science. And first of all, I would like to say that my first love was with animal behavior. In particular, massive changes that happened in the study of animal behavior in the 1960s and the 1970s. Before this period, people working in this field had no genuine theory. They were working on different species, different groups, and they had little in common. And in the 1960s, well, there has been a flow of ecological and evolutionary theories that brought some unity. Whichever your organism, you could contribute to common issues. And my PhD was on Bill Hamilton, the most creative evolutionary biologist of the 20th century, the man who was said to have solved the problem of altruism, evolution, solved the problem of the origin of social evolution in bees or wasps and ants. It was more complicated. He rather made altruism an important subject for evolutionary theory. His work led to major research programs on animal behavior and social evolution. And I mentioned him because Hamilton had ties with Brazil. After his PhD, he did his first postdoc in Brazil with Warwick Care, precisely to test his ideas on the evolution of social insects. And Brazil was then a privileged place for studying primitively social insects, these species that were partly solitary and partly social, and that were thought to be the key to the transition to social life. So another famous scientist was the American bee expert, Charles Michener, who conducted observations in the state of Paraná in the 1950s with Rudolf Bruno Langer. Then other countries took over. So Mary Jane Westerberg settled in Colombia, Raghavan made studies in India. But at one point, for those interested in the origin of social insects, Brazil was the place to go. And we have no history of these expeditions. So this is possible. But the first thing I would like to explore in greater detail today, for Joao's instruction, is the history of ecology. Because this is something I started to work on the subject when I was in Brazil. I started to work on it, then I had to leave Brazil, and so I stopped working on the subject. Now that I am back, wow, maybe I can start again and be 10 years younger. So, population and community ecology, it's a field that benefited from intense mathematical modeling since at least the 1930s. But the application of mathematics has encountered great difficulties. Most often, the mathematical models that were devised in the early 20th century were focused on simple interaction between one prey and one predator, or two competing species. And we know that in nature, species interact with many other species. So the applicability of these models, mathematics to nature, has been a fundamental problem. So there was this major gap between mathematics and nature. But there was another gap in ecology, and which the historian of science, Robert Kohler, has popularized, illustrated. It's a fundamental distinction in biology, in the tradition of field studies and the tradition of the laboratory. So I don't know if you are familiar with this book. So it's a really beautiful study of the communities of naturalists of the late 19th century, North American scientists mostly. And Kohler reminds us that the 19th century was a triumph of laboratory sciences. If you wanted to do science, genuine science, you had to do lab work. And this was felt as a challenge by the zoologists, the botanists, who wanted to work in the field, who wanted to study nature itself. But it was considered as descriptive work, below the standards set by the lab. And Kohler showed how several generations of naturalists tried to answer these challenges by importing lab standards in the field to make studies more quantitative, to devise experiments, to make their science respectable. And ecology in this geographical area, it was defined as scientific natural history. This is the definition that Charles Elton gave. It's natural history made scientific. And what's beautiful in this book is that Kohler does not simply chart the ambition, but it shows how difficult it was for these scientists to raise standards and to try to emulate lab scientists. And it shows the disruptive effects of these ambitions. There was a whole generation of naturalists who stopped doing work because they could simply not be at the level of their standards. And more from a methodological point of view, Kohler what he did was a study of traditions, how traditions are in competition between themselves, but also how they can interbreed, mix, hybridize, and how you can colonize the border between these two traditions. So what color raised as an issue is the relations between research traditions in biology. So you can be interested in relations between theories, generalizations, but also relations between research traditions. And it seemed to me that we could broaden the scope of color study by taking into account the interaction with this third area, this third tradition, that of mathematical model building in ecology. To what extent did models circulate between the labs and the fields? To what extent could naturalists want to know about mathematical models? Right? Nobody was trained to understand them. To what extent could you apply mathematical models to nature itself? And on this issue, there is a wonderful paper that was published in a philosophical journal, Synthesis, by two ecologists, two laboratory ecologists, who devised a beautiful narrative on the history between the lab, the field, and the maths in the 20th century. And it's a narrative full of nostalgia, because for them, the 1930s up to the 1950s was a golden age of laboratory ecology. Why? Because they considered that the laboratory became the medium between the maths and between the field. Nature was too complicated to apply mathematical models, but in the lab, you could do that. You could study phenomena by controlling the phenomena you wanted to study. And the canonical example in the narrative is Gauss's set of experiments which he devised to get postdoctoral grant in the United States. So it's the best application project that's been ever submitted. It was rejected or it's failed. But we still have the science. So maybe applications are a good thing after all. And remember that Gauss devised many experiments. One of them was attempting to make two species of Paramecia coexist in tubes, and he failed until he managed to provide two different sources of food for these two species. And he concluded that everything else being equal, coexistence should not exist. Competitive exclusion should be the expected outcome unless coexisting species manage to reduce interspecific competition by using different resources or by exploiting different niches. And these experiments were influential enough for prompting naturalists, notably in the United States, United Kingdom, to study differences in lifestyles and provisioning to interpret biodiversity as a result of niche partitioning, niche differentiation, so as to limit interspecific competition. These are tits, British tits, who coexist in any European forest. And this was the result, according to very painstaking observations. This results from the fact that they exploit different levels of the vegetation. So there was this wonderful division of the world, this Yalta partition. Mathematics say what is possible, the lab shows how it is possible, and the naturalists try to check, to confirm it with the field studies. And Merton, Macaulay, these guys, they felt that this happy state of things was broken in the 1960s when Robert MacArthur and his followers decided to bypass the lab and to build mathematical models straight from field studies. For many naturalists, MacArthur has been extremely inspiring because he gave the feeling that all the major questions of ecology could be asked and could be answered with models that could be applied to nature. And so there is a very nice history of ecology, also by Charles Kingsland. It's the history of theoretical ecology, which is focused on MacArthur, which is portrayed as a crowning point of the history of population biology. What is less known is that MacArthur triggered great hostility among lab researchers because they consider this use of models are metaphorical, as dangerous. Because MacArthur said, well, I think of a question, I make a model, I try to make some predictions, and I go to the field to confirm them. For lab scientists who try to produce phenomena, this was anathema. And so this shows how viewing the history in terms of relations between traditions can be very helpful. And nevertheless, MacArthur brought a breath of fresh air in this period by this feeling that you could answer the big questions. So it was very speculative, but it's fantastic to be a student and think that, wow, you can settle the problem of biodiversity. And what I studied was a group of ecologists who was inspired by MacArthur, by the idea he could ask the big questions, but they decided to use another approach. They wanted to use lab standards, but in the field. And how did they do that? By conducting field experiments in the environment of the organisms. So I was interested in some experiments in the tidal, especially on the rocky seashores in the North Pacific, and the studies of the group of Robert Bain. I was interested in it because these were considered as classic experiments, and I wanted to better understand them. I don't know if it is still taught in ecology. Are there ecology teachers? You are trained in ecology, so to mention. Paolo, do you teach that in your lectures?
Sure.
Sure. Okay. That's good.
I think so.
There's always a regret. Then I could say the things that I find really interesting, but
Let's see what to say.
Let's keep. So in a textbook way, I will mention that Payne was a naturalist who was interested in trophic interactions, trying to map the diversity of species living in a steady system, and made a footwork showing that this system was under the control by a top predator, a starfish. And Payne devised removal experiments by removing the predator from a very, very small part of the rock, a few meters. But it was some work, just to exclude the predator for 1 year. It requires some work. And in 1 year, mussels had colonized most of the rock when the predator was removed. And from that, he drew 3 consequences. I don't know if you teach it this way. The first, this shows how exciting it was, because you could draw so many theoretical consequences from a single small experiment that statistically raised each week. The first was a hypothesis on the maintenance of biodiversity. Payne concluded, well, actually, there is very strong interspecific competition for space in my study system. I could believe that many species coexist because each of them uses different resources, but no. Left to themselves, they enter in competition, and the mussels win, and they eliminate all the others. This means that biodiversity is not a natural outcome of interactions between species living at the same trophic level. Payne thought that in this kind of system, there is a very strong interspecific competition, which would lead to the extinction of biodiversity. And so if biodiversity is maintained, it's because of extraneous factors, such as predators or physical stresses, which perturb, which challenge this competition process. A second consequence was a way of studying the ecology of communities. Distinguished way of studying communities was by the use of food webs. Put all the species living somewhere and we try to link them by arrows. So this is nice. This was done in the Spitzberg because it was thought to be a simple system, few species. But how could you study that empirically? You would need hundreds of researchers to study the effects of provisioning of each species. Different approaches were devised to bypass these difficulties. Was one functional ecology. You just forget about the species, and you look at the transfer of energy or currencies between the main compartments. And then proposed another approach to focus on some key interactions that is, some species that have some major effects on the system. In other words, Payne's conclusion was that species are not all equal. Some are more important than others because they have disproportionately large effects on the structure of the communities. And by focusing on these actors, you can get a lot of information. These are the keystone species. And the first consequence, which was really part of a trend in this period, was the turn-to-field experiment. These conclusions, you could not draw them from observations. You had to do the perturbation to observe them. So there was a strong move among ecologists to give up observations and to devise experiments to identify causal factors. This was part of the trend. There were many other famous experiments of this kind. So among the classic examples, we can give Joseph Connell did this kind of experiments on interspecific competition between barnacles, Darwin species. Dan Jansen did experiments on the coevolution between acacia and ants to show the mutualism. Dan Simbolov fumigated entire islets to measure the process of recolonization by the species on the continent. And there was a very strong push for experiments in this period, to the point that one of the most famous British ecologists, John Lawton, in the early 90s, said the pendulum has moved too far away. You are not a real ecologist unless you do manipulative experiments. This is what big round-up science is about. So I think that today we have a different perception because we see that this wave of field experiments stopped in the late 1980s with the growth of microecology. Ecologists learned how to use a computer and to learn statistics and to gather a lot of data and try to infer information from the patterns. And with the emphasis on climate change, this was also something we do by field observations. And my main interest in this line of work has been the problems met by Payne and his students when they try to consolidate their results. It's nice to do one experiment that has a major impact, but how do you replicate it? Knowing that each year the conditions will be different. What happens if you do the experiments on another place or just a bit lower on the seashore? How do you deal with the variation of field condition? Is starfish keystone species only in some specific place or at specific times? And so how do you connect the seashore with the sea, which provides the propagules for the species colonizing the seashore? So it was an epistemological question. How can you stabilize phenomena under field conditions? And I think it's an issue that has been really tackled by the philosophical literature. And I think more generally, the entire history, the entire literature on ecology has not been studied by historians interested in epistemological issues. So I think there is much to do on this issue. Il me reste combien?
Ça fait 25 minutes.
And the second thing I want to mention is my current research on the history of population genetics. Here, I want to talk about muscles, see nice environments. It's much more hostile, unfortunately. I miss the beach. I was interested in population genetics and its history because of the way it has structured evolutionary thinking. You see, indeed, every debate about evolution seems to be a debate about the place of population genetics. Is it the right science for studying evolution? Is there something that population genetics misses? And what is it? Are the processes advocated by the proponents of the extended synthesis commensurable with the framework of population genetics or not? These were open questions. And so I noted that population genetics was much criticized, challenged, but we had relatively few historical work on this field. So I wanted to better understand the field, so I had to do the history. And I worked on the history of this discipline after 1945. That is before the modern synthesis. The period preceding the modern synthesis has been well studied. We know how some geneticists who knew how to do maths divided models to frame conjectures, to frame speculations on evolution. And they made it a major language for studying evolution. But this language, this framework, was devised before geneticists got a molecular understanding of the genes. Question was, well, many things happened since the Second World War. To what extent was population genetics affected or challenged by these developments? And I went to some my views or my results. And the first question was, and this is part of molecular biology. To what extent has molecular biology changed or challenged population genetics or its formalism? And surprisingly, it did not affect it very much. The formalism remains exactly the same. Was it a major deficiency? No, it's precisely because the formalism was so abstract that it could apply to very different ways of dealing with genes. Molecular biology has not changed our understanding of classical genetics. We get half of our genome from our father, the other half from our mother, and this is enough for doing population genetics. But there were other aspects, some of them were social. Population geneticists had to learn how to do molecular biology. They had to learn these methods and keep abreast of these ever-changing methods, from electrophoresis for detecting enzyme variation up to genomics. And for studying that, I took a laboratory as a field of investigation because I thought we cannot understand science if we just focus on a single scientist or classic papers. To understand science, we need to go to the lab to see how a lab works, the different communities of people working there. So not simply what the big guys publish, but what the students publish, what the technicians are doing. And the choice of Lumington was obvious because he was so influential. When was of the major proponents of molecular methods in population genetics. And his lab was always among the first to get the most up-to-date methods until the late 1980s. The paradox is that Lumington was mainly a theoretician who never devised experiments himself. And as time went by, he published less and less theory. So he had more and more data, but less and less theory. And why was it so? Because he felt that the data were unreliable. The data on enzymes are not that clean, so methods had to be improved. And then there was DNA sequencing. They had to learn how to do that when it took years to clone a gene and to sequence it. And Lumington felt that he could not make theory or speculate before he got reliable data. But it was a moving target. And so the result is that Lewinton's lab was the only one producing data on variation in genes, but the data was limited to 5 or 6 individuals, so you couldn't do population genetics, sample sizes are small. And so the goal of obtaining better data put his own lab to do very little work on the theory. And on the other hand, there was something rather ironical now Lewinton for other reasons, because in some circles, let's say the extended evolutionary synthesis is known as the advocate of the organismal view of the interaction with the environment, whereas his research program has been the most massively reductionist in the entire field of population genetics. If someone said that the progress lies in adopting molecular methods and going always closer to DNA, it is the I said that there has been no major change in the theory. It's one point of view. When we get closer, there were some trends and changes. One of them was a major transition from deterministic models to stochastic models. In the 1950s, most population geneticists thought that the deterministic factors, such as mutation, migration, and natural selection, were the prevailing factors that shaped genetic variation. So stochasticity linked to the effect of reproduction in populations of finite size were thought to be less important. And it was not a dogma. It was simply the result of the experiments, the work that was being done on chromosome variation. But with the molecules, things changed. And so my second major work has been on another laboratory, that of Moto Kimura in Japan, who fathered a very influential school of stochastic models for interpreting molecular variation. And the Kimura's group was so productive that they provided the main theory for interpreting molecular data. If you wanted to interpret the molecules, you needed to use the models devised by the naturalists. And even natural selection had to be modeled in a stochastic framework to take into account the effect of drifts. It became important to compare the strength of these two factors. And in this case too, Kimura was the main provider of models, so that provided both the models for the advocates of drift, stochasticity, and the models necessary for the study of selection in this context. So that at some point, everybody had to use either the models themselves or their results, the tests, statistical tests, to test neutrality. The third development I would like to mention is a connection between timescales. Dobrzansky's Genetics of Natural Population was a study of genetic variation within a species. He was interested in divergence, differentiation between species, but he had to rely on other methods. There was no way of connecting the study of variation within populations and the process of the origin of species. And the major change provided by DNA was that it provided a metric for both factors. With DNA, geneticists became able to compare variation and divergence, and to devise tests of neutrality, tests to detect natural selection based on this information. So the theory underlying these tests, it's an extension of Kimura's neutral theory, but some of these tests were devised by selectionists, the Lewinton students and followers, who wanted to detect selection. And one of the mastermind of this test was Martin Kreitmann. So Kreitmann was the first to sequence a sample of DNA sequences for a gene. So it's the hydrogenase locus in Drosophila melanogaster. And he thought that the variation he observed in his study was neutral. But he decided to test it. For that, he built two tests to interpret it. Which led him to conclude that these systems were under natural selection. And these tests were based by comparing variation within a species with the genetic variation in a related species. And I would like to make a comment on Kreitmann's career path, because it is often said that there has been no interaction between population genetics and developmental biology. Population geneticists pay no attention to development. And Kreitmann offers a remarkable example of developmental population genetics. In Chicago, he became interested in the development of Drosophila, the genes affecting the early development of Drosophila embryo, and he applied his neutrality tests to these very genes that control the onset of differentiation. And he did not stop to apply statistical tests. He devised experiments to substitute gene sequences from related species to test the effect of the genes. So population geneticists have been able to, by learning molecular biology, have been able to intervene and to control the information given by statistics. And DNA data has led to something else, the generalization, a genealogical approach to population genetics. The approach consists in attempting to reconstruct the genealogy underlying the genetic variation in a sample. And the mathematical framework is provided by coalition theory, which is a probabilistic treatment for describing these genealogies. So je sais pas si je vais combien de temps?
5 minutes maximum.
5 minutes maximum. So I gave 3 examples of genealogy, mainly to illustrate how population geneticists becoming interested in time and taking time into account when interpreting data on standing genetic variation. And here we have the gene genealogy, which is that of a neutral gene evolving neutrally simply under the effect of drift. And here we have the genealogy, which goes much farther back to the past. It can go even farther than the differentiation between species. And this is what is called an ancestral polymorphism, when you get genetic variation within the species, but which predates the differentiation between species, and only one factor is known to preserve this kind of variation for hundreds of thousands of generations, which is natural selection. Natural selection maintaining variation, so balanced natural selection. And this is an example of very recent contraction of the genealogy. All the members of the sample are closely related, are different, but the mutations are originate from a recent origin. And various things can cause this kind of pattern, including a recent episode of selection. Yep. And so this is something that was completely unthinkable in the 1950s, that in the population genetics lab, trees, branch length, shape of the genealogies became the defining objects of investigation. And this is linked to Warren Ewan's formula for describing these changes. So Warren Ewans was a major mathematical population geneticist who, in the late 1970s, decided to switch subjects, and he decided to write a textbook, to close a book on what he knew. And he summarized his vision of the history of the discipline. He was a scientist giving a history of the field. And he explained that the focus of population genetics had changed. Previously, there were little data, very few data, and population genetics tried to predict evolution, what was most likely to happen, knowing that there were no data for drawing inferences. And with molecular methods, population geneticists gained access to this data. The role of theory changed. It was not anymore predicting what should happen or what would happen most of the time, but it became aimed at interpreting the data, and that is identifying the factors that affected the history of a species or of a sample. And this is what Juven's called the transition from the prospective to the retrospective discipline. So population genetics became a historical science.
Thank you. So there's a lot of material to discuss. Who wants to start for the discussion? Yeah.
You said that you start your interest on science considering animal behavior, and you start your presentation with a Hamilton proposal that involves how it's possible to be able something like a large ant colony, or bee or basically. But when we consider the idea, it's a question, it's not a provocation or not more than that. When we consider the idea of Hamilton, he used a model to propose the mathematical solution that's based in the fact that the queen had only one male involved in the process. But the point is, when we look for, and I work with ants like Atta, called foraging, leaf cutting ants, you probably have more than one male. And that's a strong problem that's never been totally solved, because if the solution is based on the presence of one male, the presence of two males could be a strong problem, considering the mathematics involved in the relation, the genetic
relatedness between the
yeah. How you see this kind of aspects, considering the genetic aspects involved and the possible solutions for that? It's a truly question. It's not
So Hamilton devised this scheme when he was in London, and then he went to Brazil to study species in which indeed either there were several females cooperated in building the nest, and one of them later took control of the nest. And the other situation was mating by the queens. So he discussed these difficulties at length in 1964. So these were not major objections discovered later. He dealt with them in his paper. So I have not looked at the problem for quite some time, and this is why I said that Hamilton did not solve the problem of acquisition or bisociality. He opened a research program to understand why bisociality exists. It seemed to me that the best guide for this kind of issues, not with ants, but rather in wasps. And I'm trying to remember, very far away. You depend on the history of, let's say, on the demography of your species. That is, if you have if your population is strongly inbred, you see you have limited gene flow from neighboring population, you can raise kinship not by relatedness, but by inbreeding. So I think that was a possibility. That was something John Strassman, she worked on that in Mexico. And then I think it seems to me that the most interesting work is in the field of social policy. That is how the workers control what the other members of the nest are doing, depending upon coefficients of relatedness. And I think there are predictions on the behavior of the workers depending upon the level of multiple mating by the queen. And so you can do control experiments in which you remove the queen, for instance, and you see what happens. So this is a fruitful line of work, it seems to me. So I do certainly not pretend to say that this is the solution or the solution, because I'm a historian, and I have no particular take on this. But it seems to me that this has got quite robust work. So you are more familiar than me with the field?
The problem is, I'm not sure if someone make the mapping of the genetics present in a large colony, exactly what's happening during all the time of life of this colony. Because we can think different methods to control that. Probably you can have with one queen, only one queen, a large colony, and they use some space separation. That could be something like that, and kind of have two parts of the colony. You can maybe think about the division of labor. There is some group makes think about the group, and we found that on the literature that exists. But the point is, maybe the solution that he proposed, it's not exactly what is happening. There is maybe you can have both the genetic the different genetically variation, and that could be able, not considering this very simple model that he thinks, but maybe another way to organize. I'm not sure, but
yes,
we can discuss that.
Yes, well, one should be open-minded. This being said, there are some proxies for determining who is in power, for instance, in the economy, and sex ratio is a proxy because the queen and the workers do not have exactly the same genetic interests depending on the number of mating by the queen and so on. And so this is the proxy that gives information on what is going on. The problem is that it's quite difficult to study because even if you can count the sex ratio, it changes with
What do you think about the idea that, it's not a question also, but the idea that you have a superorganism that can be looked at, that can work in a different ways? That is, it's not about only the genetic perspective locally, but you have the entire colony working as an organism. And the selection is only on the queen, based on the model. There is a very different idea from the Hamilton proposal.
Yes. I have neither weak nor strong views on this. It's just that even in a state in which you assume, okay, there's a superorganisms, sometimes when you remove the queen, basic disruption appears with the conflicts of interest members of the nest. So I think there still is social control within the nest, are still conflicts of interest within the nest. So the question of the superorganism is how are these conflicts managed or resolved? And so the superorganism is not a match. It's just, you see, I just speak as an armchair historian.
For the sake of time, I should just say that if one of the proposals was to convince me that it is interesting, it has done right. So it's interesting. After, I would have some other questions.
Okay, we still have time for one or maybe two very short questions. Don't go, stay. Yeah?
Well, thank you very much for the talk. I was glad to see a talk about ecology, then I realized that only half of
Sorry.
So I will back to the first half. Well, as you certainly know, the idea of field experiment is still alive in ecology, and very well alive. I mean, a lot of people, extend the whole career doing field experiments. So I have two questions. The first one, I learned with your talk that the idea of field experiment was coined to solve the problem that lab experiments are too simplistic, but the nature itself is too complicated. So ecologists manage to find something in the middle of this. So my questions are, do you have any information, do you have any study on how specific is this solution to ecology, or can we find this very same reasoning to justify field experiments in other areas of knowledge? And the other question is, if people that are doing field experiments right now in ecology, do they use the same reasoning to justify the work, or something evolved in the idea of field ecologists? I'm not sure if you approach these problems. I'm just curious if you have some information.
Usually in physics, there is no big issue with working in a lab, because this is precisely where you can do some good physical work. You need to control your environment to extract signals. So that's whereas it's interesting that when a physicist starts to learn biology and think, yes, I would like to work on this subject, I propose this experiment. Usually, the reviewer, biology reviewer, will say the experiment is too simple. And the physicist replies very precisely, it will be very simple so that you can measure it and you can apply the theory. That's the point. If you want to get complexity itself, you cannot do serious work. And so I always find very interesting the information given by physicists to switch careers, because they have very interesting way of seeing biology, and yes, there are some cultural differences of outlook. So I think this would be a good comparison point. I'm not aware of similar. You can find it in evolutionary biology, let's say, with these beautiful experiments by the Lansky group of experimental evolution in bacteria, which you can do paleontology at the stage of the lifetime of an investigator. And you can, I think these experiments have been well received, but you can always find an ecologist who says, well, it happens in a lab, but it's a problem in social evolution, for instance. There are some people who are doing social evolution in bacteria, but it's in the lab, in an environment in which bacteria have not evolved. So what kind of inferences can you draw from these experiments? But it is still close to ecology. Lansky was trained as an ecologist, and in a circle of people who did field experiments. So he was very interested in this standard. And the second question is, if you want, maybe we can talk about it.
Maybe we don't have time for.
No, we have to stop there to keep the schedule, but we will have time during the coffee break.
Sure.
Okay, thank you, Baptiste.
Thank you.
Okay, so before the break, we will have our second speaker for this afternoon, and I don't